All right, this is part 2, continued in chapter 43. Inflammation. So inflammation is bringing in additional fluid and cells to a site of damage or infection to further defend oneself. It's triggered by the release of histamine and other chemicals. And essentially what happens is blood vessels dilate, they expand in size, and bringing in more phagocytes to the scene, and also antimicrobial proteins like the proteins of the protein complement system. And they enter tissues. This also causes capillaries to become more leaky, which we'll show you here. Pathogen enters. Here's the blood vessel. You'll notice it gets dilated. Proteins and white blood cells here pass through, and they pass through because the vessel becomes more leaky. And they know that the cells are attracted to the area of infection because these chemicals here, and these blue dots, are released from the cells that are damaged. And these chemicals are called chemokines. And chemokines attract phagocytic cells from the blood. And then here you see the phagocytes going to work. These are probably macrophages, but also neutrophils you see here. And they consume these pathogens, and we'll talk about how and why that works later. NK cells, or natural killer cells, these float around and look for cells within your body that have been infected with a virus or are identified as cancerous. So misprogrammed, bad cells that need to be killed. They're targeted for death. You recruit and bring over natural killer cells to attack those cells. Sometimes it can be difficult to identify cells of your own that are attacked by, or that have been attacked by virus, and the viral particles inside. The viral genome is inside the cell. But this is what natural killer cells do, and then they trigger apoptosis, or cell death, in the cells. Now, sometimes natural killer cells can go haywire, and they can cause cell death of your own actual cells that we call self, not non-self cells, as cancer cells sort of have become, and so on. But that can be a problem, of course. Autoimmune diseases like that. Invertebrates also have immune systems. They use same mechanisms as the vertebrate innate response, same idea, preventing things from getting in and so on. And even plants, as we did in class, that activity, plants have some of the similar types of ideas for their innate immune system. All right, acquired immunity. This is now very specific defense against infection, and it's tied to lymphocytes that we'll get to. Acquired immunity is the second major kind of defense, lymphocytes. Now, lymphocytes are going to respond to antigens. Now, antigens are foreign molecules of any type, even if it's something you shouldn't be viewing as foreign, like pollen. If you view it as foreign, we essentially group it and call it the proteins on that an antigen. And these antigens are going to, you're going to produce a specific response to them using lymphocytes. So lymphocyte doesn't recognize the full antigen. Here we have an antigen in yellow. And notice the full antigen doesn't get bound by proteins in the body, only the tips of the antigen. And these tips we call epitopes. Epitopes can change or vary, and this is the thing that's going to be sticking out. Actually, the full antigen is going to stick out, but the epitopes will be bound by antibodies or other structures to kill them off. Now, there's two types of lymphocytes in the body. Lymphocytes include B cells and T cells, these two categories, and they circulate through the blood. The plasma membranes of both B cells and T cells have about 100,000 antigens that all recognize the same epitope on them. So here you can see in this picture, here's a B cell. We're only showing you a few, but here's a bunch of different antibodies, essentially, sticking out of the B cell. And these recognize a specific epitope or antigen. Here you see the antigen binding site. The V stands for variable region, and the C stands for constant region. So in all B cells, the antibodies that are sticking out, these transmembrane proteins, include a constant region, and then this variable region will change, or it'll vary between all the different B cells. Sometimes instead of calling these antibodies, we call them immunoglobulins, because they are members of the globin family of proteins, and they're part of the immune system, of course, and they're going to bind to antigens. Now, later we'll talk about selection, clonal selection, and how you choose or increase or produce a bunch of antibodies. Here we have T cell receptors. This is a T cell. You'll notice once again we have a bunch of antibodies sticking out or attached to the membrane of that T cell. There's a constant region and then a variable region. And all T cell, this one T cell has all the same antibodies wrapped around it, attached to it. Actually, they're not called antibodies in this case, but never mind. Now, T cells work in a different way from B cells. They still bind to the small fragments of antigens, but they use MHC molecules. These small fragments of antigens, a cell will use an MHC molecule to expose or to bring forward the antigen. So the small fragments of antigens that are bound to normal cell surface proteins are called MHC molecules. And MHC stands for Major Histocompatibility Complex. Now, infected cells, such as, let's say, a skin cell that's been infected by a virus, has MHC molecules inside. And those MHC molecules are going to bind to the antigens of that virus. And then they're going to transport those antigens to the surface of the cell. And we call this antigen presentation. Okay? Now, there's two different types that we're going to show you or talk about. One associated with phagocytic cells, like macrophages, dendritic cells, and so on. And one associated with body cells, other cells of the body that have been infected. The important part of doing this antigen presentation is that the antigens are now being shown on the surface of the cell to the immune system for identification and then removal later. And these MHC molecules, different ones work with different antigens, but that's okay. So here we have class 1 MHC molecules, and this is what I was just mentioning previously. All nucleated cells of the body use class 1 MHC molecules. Class 1 MHC molecules work with T cells, as you can see there. Here the antigen fragment is bound to this MHC molecule. It's transported to the surface. And then the class 1 MHC molecule shows this antigen, essentially, to this cytotoxic T cell. Okay? This cytotoxic T cell will now know which antigen has infected the body or is found in the body. And this one that binds to this antigen will undergo clonal selection, or a complicated way of saying you make a whole crap load of these killer T cells. There's also class 2 MHC molecules. Class 2 are found in dendritic cells, macrophages, and B cells, those three. And they communicate with helper T cells. So here we have a class 2 MHC molecule. This is a phagocyte. It consumes the pathogen, breaks it down, and the antigens from that pathogen are bound to a class 2 MHC, taken to the surface. This antigen is shown to a helper T cell. The helper T cell you'll see later is like the conductor of the orchestra. It tells everybody what to do. Or if you like, it's the operator that you call when you call 911, and they can communicate with ambulance or fire or police or SWAT or whatever. And then an immune response will be started specific to this antigen. Now, production of lymphocytes. So they come from the bone marrow. They all start in the bone marrow. And where they mature is how they are defined or their named. So newly formed lymphocytes here start in bone marrow from stem cells. They're called hematopoietic stem cells. They originate in the blood. They're also the same stem cells, for example, that when you are born, if you had your umbilical cord cells collected, those cells are hematopoietic stem cells, so they can become any of the blood cells in your body. They're also made in your bone marrow, your red marrow within your body. If these cells mature in the bone marrow, then they become B cells. If they mature in the thymus, this is a gland, an organ that sits right on top of your heart. If they mature in the thymus, then they become T cells. Notice T for thymus or B for bone marrow. Now, as they're maturing in the bone marrow and the thymus, the receptors on these cells are tested for self-reactivity. So you run through this check and make sure that these receptors aren't going to bind to any of your own proteins and attack your own body to determine self from non-self. Sometimes this checking mechanism doesn't work, of course, and we get autoimmune diseases, such as in diabetes or multiple sclerosis. And then any lymphocytes that bear receptors for self-antigens are ideally destroyed, right? So here we talk about that clonal selection that I mentioned. Once you identify which cell has the proper receptors to bind to the antigen, it will be cloned and made a whole ton of. Here we see 3 different possible B cells with different receptors, right? Different variable regions on those receptors. Antigen binds to this one, so we want to clone this one up, make a whole ton of it. Here we have a clone, and these cells are going to mature into plasma cells, and once they're called plasma cells, they release their receptors, and these little things that have been released are antibodies. And then you also keep a small subset of those B cells that have been cloned to remember the infection for next time, and we call these memory B cells. So the second time you get exposed, you can elicit a response. Here we have, for example, an initial infection at day 6. You produce antibodies to it over time at about day 14. They're peaking, 15 peaking. Levels fall as the infection disappears. Now you've been exposed again at day 30. You can see the slope of the line and the amount, the antibody concentration is significantly higher the second time around. This second exposure you don't even know you've had. You kill it off extremely quickly. And then you maintain fairly high levels for a period of time of that antibody to ensure you don't get infected again. If you're exposed to a different pathogen, such as pathogen B, then you would create new antibodies as you did before. And this is the basis for vaccines, by the way. And we'll take a pause here.